afternoon, and this is going to be the pre-lab for your next lab, which is acoelomate and pseudocoelomate animals, meaning lower animals, those without a backbone, invertebrates, but not. These are like the lowest of the low. Then there's the mid-grade one, which have a coelom. Um, if you looked at the first PowerPoint, what coelomate means, a coelom is a true cavity inside of an organism, meaning that there has to be three germ layers of tissue called ectoderm, mesoderm, and endoderm. Ectoderm is what's going to make the outside epithelial, endoderm is going to make the inner lining, and then the mesoderm makes up usually the bulk of the organism, like all the muscle tissues, most of the organ systems, if they have them, which would make the skeletal system if they had them, and so on and so forth. So, whenever you have acoelomate means that the creature has three embryonic germ layers, ecto, meso, endo, and they're all stacked up on top of each other with no gap in between them. That gap is important because that gap allows a space for more advanced organs to form and more systems to form. So whenever you're acoelomate, you can't have an advanced organ system because there's no room. When you go pseudocoelomate means there's a cavity or a space between the ectoderm coming into where the mesoderm is, but the endoderm is not fully lined with mesoderm, meaning it's not a true cavity. These would be different types of creatures that hopefully you'll see in the PowerPoint, like nematodes, which are roundworms, like Ascaris, which is one of the things I'm going to draw, as well as rotifers, another thing I'm going to draw. When you get to coelomate's called a tube within a tube body plan, which means now you have all three germ layers, you have an endoderm, and it's fully lined with mesoderm, and there's a distinct cavity. That cavity is what you would call things like like your thoracic cavity, abdominal cavity, your pelvic, that allows space in order for more advanced systems to form. Okay, at this level, don't have any of that, really. So we're not going to have anything that you would call, kidneys, nothing. There's no gallbladder, none of that stuff. Okay. Most of them, if you're lucky, you're going to have what's a gut, basically an intestinal area, a mouth, a pharynx, which is your throat, no jaws. You're going to have gonads. And instead of having a kidney, you're going to have a water balance system, which is either going to be when we do the jellies, water balance is just done through osmosis, high to low concentration. There is no excretory system or circulatory system or respiratory system. Once you get to further up creatures, their excretory system, how they get rid of waste, is usually for osmobalance. At the very primitive layer, once we get to flatworms, that's going to be flame cells, which then would evolve more advanced into what we're going to call nephridia, nephrostomes, which then will evolve a little further into what will then become, we'll call them green glands and Malpighian tubules, which are hop, skipping, and jumping away to kidneys. Okay, so it's an evolution of the excretory system. At this level, once we're talking about jellies and the smaller creatures, basically it's just water flows in, water flows out is how it works until we get to flatworms. All right, so here we go. In your pre-lab, the first thing you have to draw is, label is going to be a sponge. Okay, so the sponge is going to be called Grantia. Now, when you think of a sponge, this is not how it's going to look in your lab. When you think of a sponge, think of a chimney. Okay. So basically we have an opening, we have a tube, and then we're going to have tissue surrounding it, okay? This part right here, that's what you would call the actual sponge, which is what you would use in the shower, okay? It's made of a series of cells called mesohyl. It's not a true tissue. However, there's an outside epithelium, there's an endoderm as well too, but there's really no true tissues in a sponge. Now, also in this area, you're going to have structural components called spicules. Spicules are either made of calcium carbonate or glass, silicon dioxide, and that basically has a framework to hold the thing together. Now, in order for this to feed, it's a filter feeder. What's it going to do? It's going to suck in water from the outside, filter it, and then it will push that filtered water outside the middle. Kind of like a thing of like a filtering system, like a chimney. Now, all along these little holes right here, those little holes are called ostia. That's the pores or the holes of a sponge. They are aligned with these little tiny cells called collar cells or choanocytes. Those are the things that I said in the previous lectures with Protista, choanoflagellatia, which gave rise to animals, colonial flagellates. As they beat, they create a current which sucks the water in. Well, what filters it are other cells that are going to be found in the mesohyl called amoebocytes. They're like amoeba-like cells, but when they digest the material, instead of using it for themselves, they disperse it to all the other tissues. Kind of like a very, 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 very low primitive digestive system. When we come in here in the middle, this middle area is called the spongocoel. Seal is, again, a Latin word meaning cavity. It's the cavity of the sponge. When water comes out, it comes out through this hole in the top called the osculum. All right? Now, when you see Grantia, what you got to imagine is, think of the sponge kind of like a cylinder, but now you're taking the cylinder, tilting it toward you, and chopping it down. That's what you're going to see. So instead of seeing it this way, lengthwise, you're going to see it's going to kind of look like this. Okay? Understand there's going to be a hole in the middle. That's the osculum. That's the middle. These little invaginations, each time it invaginates in, that's a hole in the sponge. That's an ostium, a pore. And when you look at it, you'll see a bunch of little dots lining each one of these invaginations. Those are the collar cells that would beat to suck the water in. You might see some remnant tissue in here. Remnant tissue would be the mesohyl is what you would see. The label. All right. Next slide is going to be what are called scypha spicules. Nothing to label there as you see in the lab. Basically what I want you to see is the spicules look like little tiny glass rods or pins because that's essentially what they are. They give the sponge its structure. Next up then, we move forward and we go into the stinging jellies. The stinging jellies are called cnidarians. Cnidarians, the C is silent. What do they have? What makes them stinging jellies are called cnidoblasts or nematocysts. They kind of look like little small protein harpoons that will shoot out under hydraulic pressure, and they're dipped in usually a neurotoxin venom when they impale their prey. This would include everything from, hydrozoa, which would be hydras and fire corals. You would include things like cubozoa, box jellies, scyphozoa, which would be what you think of when I say the word jellyfish. And then, I'm forgetting one. Brain fart. That would be the anemones and the hard corals. Cubozoa, scyphozoa, anthozoa. Anthozoa is going to be the hard corals and the sea urchin. Not sea urchin. Sea anemone type organisms. Okay, so next drawing is Hydra with a bud. Now this is from the group Hydrozoa. So every time you see these organisms, understand can exist in two life forms. A polyp and a medusa. Some of the classes can alternate between both. Some of them only stay in one versus the other. The polyp stage is used basically for asexual and sexual reproduction. It's stationary. The medusa stage, that's the mobile stage, and that's a sexually reproducing body. All right, so the hydra, when I say a feeding polyp, that's the attached stage. Okay, so when I say the hydra with bud, what you'll see is kind of like a tube coming out like this. What you'll then see are a bunch of little tentacles coming out, and that's where it's going to have the nematocysts, the stinging cells. You'll see a little hole right there. That's the mouth. And then it goes in this little cavity right there. That is called the gastroderm. Basically, it's not even... these creatures have two layers. They have an outside ectoderm and an outside endoderm. They don't have a layer in between. It's called mesoglia, so they don't have the three tissue layers. But that's the gastroderm. Essentially what you consider like a primitive stomach is what you would do. Okay, now, coming off of it then, what you would see is kind of like this weird little mini-me budding off the side of it. That's the bud. It's an asexual reproduction. Imagine you get to a certain size, and all of a sudden off your hip comes a bud, and then it breaks off and makes a new you. That's that. Okay, it would be genetically identical to it. Next one you see there, it says, the hydroid colony. Basically with this, what you would see is a bunch of these kind of like feeding polyps all over the place, like this. And then every so often, what you might see is coming off of it kind of like a tube with these round little cells in it. That's a sexually reproducing body, okay? So you have the feeding polyps, and then you have the sexually reproducing area. Next one would be the Obelia medusa. Now when I say the word medusa, understand this would mean the same thing for Obelia, the hydra. Also, when we do Aurelia, which is the jellyfish, when you think of a moon jelly, like the big ones, that's what this looks like. Okay, so here we go. For a jelly, in any case, any medusa, what you're going to have is a dome. The dome is called the bell. Then you have this ring around the bottom of it. That's called the neural ring. That's where you're going to have a loose, sparse arrangement of neurons going in a circle. Very crude nervous system. Then how do we talk from one side to the other, or a series of nerves which would go from one side to the other. Think of it like spokes in the middle of a tire, a bike tire type of thing. That you know that you can do the neuroglia. I'm fine. You don't need to worry about that. Now, coming down off the bottom of it though is going to be the mouth. That tube leading up to the middle, that's the pharynx. Pharynx is the equivalent of what you would call throat. This would now open up into 1, 2, 3, 4 pouches. That's the gastroderm, the primitive stomach. Now, when you think of a moon jelly, when you look down on it, you see these 4 little crescent moons. That's not the gastroderm. Surrounding each of the gastroderms are these little white areas, and those are the gonads. These creatures are hermaphroditic. They both have male and female reproductive capability. Now, understand also, too, they do not have a complete digestive system. All they have is a mouth. So what they do is they bring food in, they digest whatever they can, and then everything comes back out the mouth again. Same thing with reproduction. Egg and sperm will come out of the mouth, and they'll have a mass spawn out in the water. Now, coming down off of this, obviously the tentacles, which would have the nematocysts on it. Okay? Now, another thing about this ring, if I were to blow it up, every so often in that ring, you would see like a little nodule. That little nodule has a little protein marble in it called a statocyst. Now, there's usually, again, it's symmetric, roughly like 8 spaced around it. How they work is like this. Think about like if you're doing carpentry and you're trying to see whether something is level, and then you have the 2 lines with the bubble. How do you know is if the bubble is in between the 2 lines that you're level. Now imagine I had a circle, like a hula hoop, and I had 8 marbles in it. Well, if I took the hula hoop and I tilted it one side, the marbles would slide to one side or to the other side. That's the statocyst, is how the jelly or the medusa is going to be able to ascertain orientation and balance. Oh, I'm tilting to the left, I'm tilting to the right, I'm upside down, I'm right side up. That's what they use. I'm trying to think of anything else that I told you to label. That should be it. All right, so next we go through the life cycle of Aurelia, which is a moon jelly, okay? All you see is in the practical, in the lab, I give you the stages. I just want you to know the stages in order and kind of what they look like, nothing to label. The first one is a planula, which looks like a little speck. Next is what's called a scyphostoma. A scyphostoma looks like a feeding polyp, same thing. Then you go to what is called a strobila. The feeding polyp, because it's attached, will stop growth, and then it will kind of look like a stack of saucers on top of one another. Each one of those pieces is called a strobila. What are they? Baby jellyfish. So what will happen is during a process called strobilation, think about taking a deck of cards and holding it and then taking each card and just wiping it one after another. Now imagine each card became a new deck of cards as it did that, or like a new jellyfish. That's what happens here. The next stage after scyphostoma is called ephra, which kind of, if you look at it, kind of looks like a star pretty much almost. It could be anywhere from 4 to 6 to 8 to 12 points to it. That's not the mature medusa. Then the last one you see there in the series of pictures is the Aurelia medusa, which is the same thing that I drew right there. Okay, that then ends the cnidarians, meaning the stinging jellies. Next group would be called Ctenophora, not in your lab, but those are called comb jellies, but I don't have any slides for you to see that. However, the ctenophorans are going to be important because they're the first ones which are going to be triploblastic, 3 germ layers, ecto, meso, endo, and even though they are jellies, they don't have stinging cells, they have sticky cells called coloblasts. Additionally, they have 8 rows of what are called tene, which are cilia. Think of like a beach ball with 8 rows of cilia that it can then move in 3-dimensional space. That's how they work. Last thing about ctenophoran, they're the most primitive creatures out there that have true muscles. First ones ever. Now, we go into the group called triploblastics, which are now going to be, first group is platyhelminthes. Platy means flat, helminthes means worm. Okay, so these are flatworms. So we're looking at everything from Turbellaria, which would be like the one you're going to draw as planaria, or the one I put out for you, you're going to label as planaria. Then you have lovely groups of parasites, like different types of flukes, and then we go into Tinea, which is going to be tapeworms. All right, so first one up on our little hit parade is going to be, you see there it says the fluke sporocyst. Now, I'm going to do this out of order so you can understand the life cycle, okay? What we're going to walk over is the life cycle of the Oriental liver fluke, okay? Now, let's just say you decide to eat uncooked fish flesh, okay? That uncooked fish flesh, if it was derived from tainted waters, could have gotten the Oriental liver fluke infected in it, okay? Now what happens is when it happens, the fish's operculum opens and a stage, which we're going to come into, will swim into it and bury itself into the flesh, into the gills, and then into the bloodstream. It'll then go dormant inside of the fish flesh, okay? So I'm going to pick it up with where you ate it if you didn't cook your food, if you didn't cook the fish. So what you end up swallowing is going to be what is called a Myrichidia stage. Nothing to drop. Not Myrichidia, metacarcharia stage. The metacarcharia stage is one of the stages you see there, and what you see, it kind of looks like a little dormant cell with this cyst coat around it, and that's really all it is. It's a tiny speck. You wouldn't know that you ate it. Why is that cyst coat on it? Because when you swallow it, your stomach acid will eliminate the cyst coat, and now this metacarcharia stage will become viable in your intestinal tract. What it will then do is it'll burrow through your intestinal tract, going into your bloodstream and then going into the bottom of your esophagus. Go in your bloodstream and then travel in your bloodstream to go to the liver, hence why it's called a liver fluke. Why the liver? Because the liver is this smorgasbord of blood that is a filter, and therefore that's like the holy grail for the liver fluke. Now, the one thing I need you to label and know is the adult stage. So the adult stage is the flatworm. It looks like this long, skinny thing. You're going to have a mouth up at top that usually has a sucker and usually small hooks in it. It'll then come down to a small little muscular area, the pharynx again, the throat essentially, and now the intestines will fork left and right. So they have two intestines. Notice it's not a complete digestive tract. You don't have an anus, all you have is a mouth. Now, more importantly, again, understand you all the epidermis and muscle tissue in here and everything else. Now, important thing is, in the middle part right here, it'll look like long black strands, like long dark spaghetti noodles. That's the uterus. The reason why it looks dark is because those are fertilized eggs on the inside. Directly behind it, you will see a small pink fluffy, what kind of looks like a donut. That was the ovary. Behind that then, you would see a series of more spaghetti noodles, but they would be very lightly colored pink. Those are the testes. Again, hermaphroditic, both male and female. Both are functional. Now that's what would be in you drinking your blood in the liver. Now follow. Eventually, your immune system kicks in and your temperature kicks up and you start an immune response. From the perspective of the fluke, its environment is going badly. It's got to get out of Dodge. So how does it get out of Dodge? It's got to make a mobile sexual stage called the Myrichidia. Okay? The Myrichidia kind of looks just like the metacarcharia, almost, very similar to it. But this is essentially what you would defecate out whenever you're infected. Now, how it does it, this is the cool part. When this thing is ready to die, it'll burrow in the bottom of your esophagus, climb up the back of your esophagus with its sucker. On one end, take its sucker, lodge it to one side of your esophagus, and then lay its eggs in the back of your esophagus. What it'll then do is with the back of its tail, it'll then reach out and lightly flick the back of your esophagus. When you feel that sensation, what do? You go, and you swallow. This just committed suicide. It's going to die in the hydrochloric acid of the stomach, but now the Myrichidia have entered into the stomach, and you're going to defecate them out into the environment through your feces. Okay? That's the Myrichidia stage. Now, this becomes more prolific in areas where you think of, quote, like third world countries, where maybe that body of water that runs past your village, yes, you used to bathe in, to draw water from, to do everything, including urinate and defecate. So therefore, if you defecated into this, you would release the Myrichidia into the water. What now happens is the Myrichidia is looking for its next host. The next host is a snail. Either the snail will ingest it, or the Myrichidia will get it in other ways. That's the next stage you see in the that's the first picture you see in the series, called the sporocyst stage. What you're looking at is a cross section of a snail's body, like chopped straight down, and what you'll see is light pink fluffy tissue. That's normal healthy muscle tissue. But what you need to note Every so often you see these little chambers with these little dark circles in it. That's the sporocyst stage. Still inside the snail, the sporocyst stage is then gonna metamorphose into what is called the iridae stage. The iridae look like these weird kind of like bratwurst sausages. They're little small things, but then they have like these little squiggles in them. What are those? Those are maturing worms on the inside, okay? From the iridae, what hatches now is what is called the cercaria stage. The cercaria looks like a weird sperm cell with a forked tail. Okay? Like that. Okay? So this hatches from this. The cercaria stage is now what's going to leave the snail, go into the water, find an unsuspecting fish, infect it, and then it will go dormant to become the metacercaria stage again. Which then, if you eat it, the whole thing starts all over. Now, why do I go through this? How many of you like eating sushi? Specifically, understand sushi is just the wet rice. I'm talking about if you were to put raw fish on it. Now, again, you're assuming the fish that is being on your sushi is been caught from untainted waters and is clean. That's a big assumption. You do understand. You do understand when you eat sushi, you're given two things. You're given wasabi and you're given ginger. If you're the type of person who doesn't eat these things, I'm hoping after this, you'll at least think twice before you go, I don't want it. Why is the wasabi there? Wasabi is a cousin of horseradish, very pungent. Anything that's super pungent usually has antibiotic qualities. What the horseradish does when you eat it with the raw fish product is it takes this cyst coat and it softens it. So therefore, when you swallow it, your stomach acid will destroy the metacercaria stage. If you don't eat the wasabi, the cyst coat stays intact, your stomach acid gets rid of it, and now the fluke becomes viable in you. Make sense? So what the hell is the ginger for? The ginger is a natural homeopathic agent to relieve nausea. If you eat something so pungent, you run the risk of upsetting your stomach. So what do? You take the ginger, it settles your stomach, and therefore you can keep it down and not get parasitized. That's the point of wasabi and ginger. Make sense? Okay. Now, that's the life cycle of the liver fluke. From there, then, so this was the adult fluke. That's the one I need you to label. Next, a cousin of this is called a planaria. Now, planaria are non-parasitic. They are free-living. They basically look like large, they're about that big when they get full-grown and about really small. They basically they're the poster child of studies that we're working now on how to do regenerative medicine, like how to grow back tissue, how to basically do tissue culture or have tissues grow together to make viable tissue from that point on. So when you think about people now making, hopefully you've seen like where they've had a scaffolding of a lung and they've grown lung tissue on it. The beginnings of this is working with the planarian back in Nazi Germany, okay? Now, the planaria, very simple anatomy on the outside. So basically, they kind of almost look like an arrow. Okay? Now, they have two eye spots, and they look like their eyes are crossed. Okay? These eyes only see light and dark. They can't see detail, can't see motion, can't see color. These things live in freshwater ecosystems like a retention pond in the deep, murky, mucky part of the pond. They're basically detritivores, which is a nice way to say, poop eaters. They're scavengers. They whatever they can find, they'll go after. Okay, so they don't want to be anywhere near light. Okay, now if you take the organism and flip it upside down, you would think, oh, the eye spots are there. The mouth should be right underneath the eyes on the other side, like yours. No, this creature has an incomplete digestive tract. Imagine that you don't have a mouth. So you have a mouth, but you don't have a complete digestive tract, mouth to anus. Well, how could you feed you from head to toe if you didn't have an anus? Because you understand when you have a mouth to an anus, that allows you to eat continually throughout the day and never back up. When you only have a mouth, you have to eat, digest, then bring it back up, which you don't then eat again. It's sporadic, it's not continual, which then limits the amount of energy you can get, which then limits how complex you can become as well too. Now, on the underside, what you would see is a tube. Basically, think where your navel would be. And there's a tube that comes out with a mouth. That tube is the pharynx. That mouth, that's the mouth. Now, when you see the hole mount of it, there's going to be this clear area in the middle of the hole mount. WM on the lab means hole mount, meaning the whole worm right there. What is that? That's the gut. Okay, that's simple as what it is. Then what you'll see is on each side, it kind of looks like two dark racing stripes on each side. The reason why they look dark is because you have three different systems, one on top of another, and you can't tell through the skin which is which. So what you have is a very crude nervous system which runs down both sides. A very crude excretory system, the flame cells for osmotic balance. And then you're going to have a very crude system for reproduction. You have the ovaries and the testes running along the side. That's why it looks like two stripes. The next series of slides then is this same worm now cut in cross section. Now there's going to be 3 or 4 pieces of worm there. I want you to focus on the middle one. The middle one's going to look like this. Oops, too much. Kind of look like that. If you cut this straight down and hold it to the side, what you will see is a hole in the middle. Well, that's the gut. Okay? You'll have the epidermis on the outside. You'll see this pink tissue surrounding it. That's going to be muscle tissue in order for the worm to move sinusoidally. Along with that then, down here in the corners, you might see 2 little holes. Those 2 little holes are the flame cells, the primitive kidney system. Now, important. Above the gut, there's going to be 2 little nodules. Those are the testes. Below the gut, there are 2 larger nodules. Those are the ovaries. Again, hermaphroditic. All right. Now, again, these things are going to breathe through their skin, plus they also have cilia on their skin to help them move as well. All right. That's the planaria. Next up, tapeworm. Good. Who doesn't love a good parasite, right? Hopefully you're not eating, anything right now. All right, so for the tapeworm, keep in mind, same thing. How do you get a tapeworm? Basically, you eat uncooked flesh or improperly cooked flesh, or you eat dirt. Pick one. People go, I don't eat dirt. If you eat vegetables that maybe you've grown or that you get somewhere else and you don't wash them, well, what do you think you're doing? You're eating dirt or potentially, so you wash it. So it's supposed to be washed. Now, a tapeworm. Tapeworm is going to have 3 major body areas. Okay, there's going to be a neck piece, a connecting piece, and then there's going to be a series of segments that run all the way back, and those are each called proglottids. The proglottids each are hermaphroditic, and they all can make separate worms, each one. It's called a tapeworm because it looks like a tape measure. If you lay it flat, the proglottids look like it's a tape measure. Okay? Now, on the top of it is the most interesting part. So these are the proglottids. That's the neck. On top is this big ball. That is called the scolex. The scolex is going to have a mouth all the way on top, a series of proteinaceous hooks surrounding it, and then a series of suckers going around the outside. How does this work? You eat the tapeworm cyst, kind of like the liver fluke. You remove the outside cyst coat, and then this becomes viable in your intestinal tract. This creature, notice, has no intestines, no gut. Why? It's living in yours and uses your intestines. So what will happen, as it goes along the inside of your intestines, it will agitate the scolex. What will now happen is these hooks will shoot out into your intestinal lining and pull the mouth onto it, and the suckers now bed in. So basically, what you would see if you were to see your intestinal tract, if you were infected, that would be buried into your intestinal wall. That would be sticking out. Now, how do you get rid of a tapeworm? You have to ask it to leave nicely. Because if you try to break it off, if you break off any of these pieces, you've now just made a bunch more worms for yourself. So instead of having one pet, you gave yourself a multitude of pets. Normally what's done is usually given, either you can be given antibiotics, but a very simple remedy they often give is a little green pill called malachite green. Malachite green, basically when you eat it, will coat the inside of your intestinal lining. It doesn't kill the tapeworm. Essentially what it does is it agitates the scolex, so it'll recede everything and let go. And then essentially you poop the tapeworm out. That's how you get rid of it, in one piece. Now, understand something. One of the largest tapeworms on record is looking around, if I remember correctly, it was something like 28 feet long. Obviously, you're not going to defecate a 28-foot long, that requires surgery, okay? Basically, the woman was in the early 1900s, and they thought she had cancer. So they went in to operate on her, thinking, we're going to pull a tumor out. And they went, no, she has a pet. But understand, in the early 1900s, they had to go through every piece of her small intestine, slowly cut it open, and take this thing out bit by bit without breaking it. Otherwise, they made a much bigger problem. She survived, which is fantastic. How do I know about this? Because this is in a big jar at the Smithsonian Institute. It's the coolest thing ever. Of course, I like weird crap like that. Anyway, so let's see. That would be Tinea. And then in the stretch, we got 3 more drawings. We got Ascaris, male and female, and Rotifer. Once we get to Ascaris, we now go into the group called Nematoda, which are roundworms. In this case now, we have a full digestive tract, mouth all the way to anus. We can have complete sex differentiation, male and female. That's going to be in the Ascaris specifically. And it's a pseudocoelomate now. It's not acoelomate. We have a cavity, but it's not fully lined with the mesoderm yet. So we can't develop super advanced systems yet. That's going to be the next pre-lab we do when we would have done dissections. And I'm going to walk you through what you should see in the dissections the next actually 3 of these pre-labs. All right, so Ascaris. So we got male, we got female, okay? Now, in both cases, I understand they're both going to have an epidermis. They're both going to have this stringy tissue coming in the middle. That's muscle tissue. That doesn't change. What you want to find in each is a landmark. That landmark is going to be this skinny little tube little thing in the middle. Now, again, sometimes it's round, sometimes it's squished. It's all a matter of when they made the slide, how they cut it. So, but that's the intestinal tract, okay? Now, how can we tell a male? Surrounding this, you're going to see a series of small little equal-sized tubes going all the way around, and they're going to have little dots in them. Those little tubes are the seminiferous tubules. Seminiferous tubules are in the testes where the male can make sperm. So all those are seminiferous tubules. The little dots inside the seminiferous tubules, those would be sperm cells. Okay? Now for the female, she doesn't have seminiferous tubules. Instead, what she's going to have is 2 very large muscular tubes underneath the gut. She is biuteral. She has 2 uteri. Understand uteri. You don't say uteruses. It's like it's that the plural is uteri. Basically, her uterus starts off and it forks to make 2 uteri. Now, those are these 2 large muscular areas. What you will then see is a series of smaller tubes kind of scattered around. Those are going to be the ovaries. The little dots inside them are the eggs. If you see dots inside the uterus, those are fertilized eggs. Okay? So for the male, all you see are circles around it, seminiferous tubules. For the female, 2 large ones, that's her 2 uteri, and then ovaries. All right? Then, last but not least, and then I show you in the lab how you can tell a male from a female from the outside. Males are smaller, they're skinnier, but how you tell a male is by its anus, it hooks. That's how he latches onto the female to procreate. The female is huge, corn-fed looking thing, and she's straight at both ends. She's bigger because she needs more energy, basically, to make the eggs. Okay. Now understand, both of these are intestinal parasites. Unfortunately, we didn't get to do the dissection, but if we did, here, imagine lo mein. That's kind of what it looks like. Or think large bean sprouts. You're welcome. I'm just trying to ruin things for you. All right, last thing is Rotifera in your lab. Rotifera has what we call a corona. Corona means that, first off, it's going to, let's see. It kind of has labial lip looking things, and then it opens up into a fat portion, and then it's going to taper coming down, okay? Now, this right here is the mouth, then we have the pharynx, and then we have the intestines coming down. Same thing I already talked about before. Now, the most important thing you're going to see about this is around the mouth, there's a series of cilia going around it. Hence why it's called Rotifera. These are called the wheeled animals. This little ring of cilia around the mouth is called the corona. Yeah, I know. Corona just in Latin means basically in a circle around, and basically the cilia, as they beat, that's how it's going to feed. It's going to bring food and basically give a toilet bowl effect, a vortex for it to be able to eat, plus also swim. Okay, a lot of rotifers when they're alive, they kind of act like an accordion. Well, they'll get really small, they get big, really small, and that's how they can move too. Now, why they're called wheeled? You look at this, you're like, how the hell? That doesn't look like a wheeled animal. Now think. This would be a rotifer laying on its side. That's this one, okay? If I were to take this creature and tilt it towards you, so you're literally looking down the mouth, all you would see is the mouth and a row of cilia around it, and as it beat in a circle, it would look like a moving, rotating wheel. That's what's called a wheeled animal, rotifera. All right, so ends the pre-lab then for acoelomate, pseudocoelomate lab. Next time I'll go over the coelomate lab 1 dissection, which would include the clam, quahog, the squid, and the earthworm. After that is coelomate 2, which would include crayfish, grasshopper, and sea star. All right, I'll see you then.